Our speaker is Dave Neitzel. He works for the Minnesota Department of Health and is the supervisor in their vector-borne disease area. He's been with the Minnesota Department of Health for 30.
Well, been with the Minnesota Department of Health for about 20 years now. But before that, I was in charge of disease prevention services at the Metropolitan Mosquito Control District. And the Metropolitan Mosquito Control folks have been doing tick studies since the 80s too. So I've been out in the field chasing ticks around since the mid-80s.
Yeah, well, terrific. He's going to, we're just really privileged to have him come tonight. He's going to give us an update on what they're doing at the Department of Health and how we can maybe work most effectively as individuals and as a group to make sure that people realize just the impact that Lyme disease is having on people here in Minnesota. So with no further ado, Dave, thank you so much for coming.
Thank you. Thanks for having me here tonight. I'm going to try and focus a little bit on what our program does and also some of the findings that we've had over the years. Like I said, I've been working on tick-borne disease here in Minnesota since 1985, back when we were first learning about Lyme disease across the nation. And like I keep on telling everybody, things have really changed since the mid-'80s. 31 years really isn't that long a time, but it's just a night and day difference, the tick-borne disease risk that we're seeing now versus then. So I want to try and give a flavor of that. I'm actually not going to talk a whole lot about Lyme disease directly. I'm going to talk a lot about the ticks. I'm going to talk about some of the other tick-borne diseases. I also want to let you know right up front too that while I've worked on tick-borne disease for a long time now, there are certain parts of it that that would be of interest to you that I don't work on. Like I don't work on research on chronic Lyme disease or anything like that. So I'm going to focus on what I do, and hopefully be some information that'll be of use to you. Also, I should mention, I know there's some specimens of ticks around the room. I brought some ticks and alcohol. For those of you that haven't seen the adult stage of the black-legged tick, what used to be called the deer tick, I have some live ones that I found on me about a day ago. So if anybody wants to see what they look like and haven't seen them, I can show you. The nymphs, didn't have any nymphs to bring tonight. They're just becoming active now, and I haven't collected any. But without further ado, I'll move on here. I supervise the vector-borne diseases unit at the Minnesota Department of Health. So when we talk about vector-borne disease, we're talking about diseases transmitted mainly by mosquitoes and ticks. And that can be a good thing, and it can be kind of a drain on resources too. Like right now, you can't turn on the TV without hearing something about Zika virus. Well, guess who's responsible for monitoring Zika virus here in Minnesota? Travelers. So it's been quite a challenge to maintain all of our tick-borne disease services, which I consider to be a lot higher priority in Minnesota or for Minnesota residents than Zika virus. So we were putting in a lot of extra hours trying to make sure that our monitoring for tick-borne disease doesn't suffer. And the main focus of our program is to monitor tick-borne diseases over time and space here in Minnesota. It's important to collect data showing how things have changed over time. And our monitoring, the methods that we use for monitoring haven't changed over time appreciably, so it's easy to kind of compare then versus now. That isn't the case in a lot of other states across the country where they've changed their monitoring methods over the years, and it's really hard to compare case numbers over time. So that's the core of our program, is monitoring these diseases over space and time. And with any resources that are available to me beyond being able to get that work done, we try and do various research projects, and I try and get out into the field. A few years ago, I was the president of the National Association of Vector-Borne Disease Control Officials, and in that group, I met with my colleagues from all over the country, and I was surprised at how few of them actually get out into the field and learn about what the risks are for either mosquito or tick-borne disease out in the field. And I don't think you can adequately understand what's going on with vector-borne disease unless you get out there and see what the ticks are doing or what the mosquitoes are doing. So a lot of our program is actually suiting up. These days we wear white painter suits, and we'll get out and actually sample ticks in various locations and I'll talk about the various research projects that are ongoing right now, but we end up testing a lot of ticks from various parts of the state. Now, when we talk about diseases transmitted by black-legged ticks, again, what used to be called deer ticks in Minnesota here, there are several different diseases of importance. The big one, of course, and the most important has always been Lyme disease. That's our most commonly reported tick-borne disease in Minnesota. The new thing that I'll talk about in a little bit here is just in the last couple of years, we've identified a new species of Borrelia that appears to be causing at least some illness in Minnesota and Wisconsin here. So that's interesting news for sure. We also have babesiosis, a parasitic disease. Human anaplasmosis, which is our second most commonly reported tick-borne disease. The agent there is a bacterium. We have a form of human ehrlichiosis, which is very closely related to anaplasmosis, and the organisms are, and cause very similar illness. And we've identified a new agent there, Ehrlichia mirus bacteria, that also appears to be causing illness here just in the upper Midwest. I'll talk about that in a few minutes. And we have a virus, Powassan virus. I can't remember if I talked about that the last time I spoke to your group, but Powassan virus is a flavivirus very closely related to West Nile virus and can cause very serious central nervous system disease. So there's plenty of reasons to be concerned about the black-legged ticks, that's for sure. Now, talking about the ticks themselves, the ticks have different life stages, and I'll go through them in detail here. And many of you have seen these ticks quite often, but the adult female will lay up to several thousand eggs on the forest floor, and they hatch into that little six-legged larva there on the bottom. So if you get a little dinky tick on yourself and feel like you can count the legs on it, if it has 6 legs, then you're looking at a larva. If it has 8 legs, it's probably either a nymph, like you see on top there, or could be the adult. And it is good to know what life stage you're dealing with, because the larvae are a lot lower disease risk than nymphs and adults. Basically, the larvae need to pick up the disease agent when they feed on an infected small mammal. Again, they have 6 legs. They're initially not born with Lyme disease bacteria. Theoretically, they're a potential vector of Powassan virus, though. The female tick might be able to pass the virus on through her eggs into the next generation of ticks. That's something that still needs a little bit of study, but we would not be surprised if there's some risk of Powassan virus transmission from the larvae. And there's a couple other diseases of importance, too, that larvae may transmit, but for the most part, they're just a pest. And usually people don't get bit by them. Mainly it's adults and nymphs that I get reported to me coming off of people. Okay, let's see. The nymph is the most important life stage from a public health standpoint. And it's not because they have the highest infection prevalence. I'll show you some numbers in a few minutes here of what we've been finding out in the field as far as infection prevalence with the different disease agents. The main reason they're so important is that nobody can see them. If you're focused on activities when you're out in the woods and brush and not looking for ticks, these little tiny ticks can very easily hide on a person. And the longer they stay attached to you, the more likely they are to transmit disease agents. So this is the most medically important life stage. They also prefer to feed on small mammals, white-footed mice, chipmunks, some of the other small mammals that are common in wooded areas in Minnesota here. And again, very difficult to detect. Now, a long time ago, I did studies in the Twin Cities here where we trapped small mammals by the thousands and removed ticks from them. And this is a pretty typical situation here. This is a white-footed mouse. And if you look closely at the ears of this mouse, you can see all sorts of little ticks on there. Those are mainly larvae, but nymphs also feed on the mice. And this is where the ticks are picking up the various disease agents. The mice and the chipmunks are the reservoir for a bunch of these different disease agents. And then if a tick is lucky enough to find a blood meal as a larva and get the energy to molt into the nymph stage, and is lucky enough to find another host as a nymph and have the energy to molt into the adult stage, The adults prefer to feed on large mammals, mainly white-tailed deer, but they'll feed on anything else, and of course they're not picky, they'll feed on people too. It's actually a lucky tick that is able to find a host to feed on at each life stage. The females, once they get onto a large mammal host, they'll take a big blood meal, and they use that blood meal to help develop the eggs that they have in their body, and depending on the quality and size of that blood meal, they can lay up to several thousand eggs. The adult males, they'll be on the same host, whether it be a deer or on a person, and they're attached to that. They'll attach, but they're not actively feeding. They're just waiting for a female to get on the same host, and then they let go and go mate with a female. So they're usually not associated with transmission of tick-borne disease agents. Again, I think Powassan virus may be something that we need to look at in a little bit more detail. But since they're not taking a big blood meal, the chance to, or the opportunity to transmit disease agents is a lot lower with them. The adult ticks, they get a couple chances to feed. They come out in the fall. If they don't find a blood meal then, they come out as soon as the snow melts in the spring. And for those of you that haven't seen the engorged black-legged ticks, adult females, this is what an engorged female looks like. I like to show this slide to physicians because everybody hears about how tiny the black-legged ticks are, but it's important to know that an engorged female is actually a pretty good size. That's about the size of a watermelon seed. And those of you that have pets and are used to pulling ticks off of your dogs, the regular American dog ticks are wood ticks. They're like a little gray marble. These things are usually a little bit flatter looking, like a watermelon seed, and kind of an orangish or kind of a dull orangish cream color. It's an easy way to tell them. Some of the research that I've done in the past has been to collect ticks off of hunter-killed white-tailed deer. I'm up at this is a slide that was taken up at St. John's University up in Stearns County many years ago. This particular deer, I was collecting blood samples from the animal as soon as the hunter gutted the animal, but I also collected ticks off of the neck, and I think I pulled 700 or 800 ticks just off the neck of that deer. The ticks are really concentrated on their host, and with the infection prevalence that we see in the ticks, that I'll talk about in a little bit here, these deer are all being exposed many times to the various tick-borne disease agents. But for the most part, they're not serving as a reservoir. They rapidly clear the agents. All you can find is antibody evidence that they've been exposed to these things in the past. But they can be used as a sentinel animal to let us know if there are ticks or tick-borne disease agents in an area. I mentioned the ticks mating on the deer. If you ever pull something like this off of hopefully your dog, not yourself, but if you, this is an adult female, it's fully engorged, and here's a male mating with that female. Again, they're not interested in feeding, they just want to let go and go mate with the female, and then after mating, the males will die, the females will lay their eggs on the forest floor, and then they'll die also. Okay, and then when it comes to black-legged tick feeding, the feeding behavior is called questing, and this is of real interest to me. With the field sampling that I do, these ticks drive me nuts because they're pretty fickle about when they come out. The tick spends almost its entire lifetime or life on the forest floor and the leaf litter. They're extremely susceptible to drying out. If we have really hot, dry, windy conditions, the ticks just stay down into the leaf litter where they can conserve their moisture. And it's days when the humidity's up a little bit higher that they can come out of the leaf litter and crawl up onto vegetation and wait for something to come by. Now, the immature ticks, the larvae and the nymphs, they'll look for a blood meal right down near ground level. They'll be within a few inches of the ground. That makes sense because they're trying to feed on small mammals, mice and animals like that. The adult black-legged ticks will crawl up a little bit higher off the ground. They're a little less susceptible to drying out, and plus, they're trying to get a little bit higher off the ground so they could get onto a large mammal host like a white-tailed deer. So oftentimes with the white suits that I wear going through the woods, I can tell instantly when the ticks get on me, and it's not unusual to get them up to about thigh height or lower. And then, of course, they walk up from there. if you have a tick on your head, it probably crawled up from ground level or somewhere down near the ground level and went upwards. These ticks do not have eyes. The black-legged ticks don't have eyes, so they can't see you coming, but they do have pretty good sensory organs in front legs and in their, well, some in their mouth parts too, but mainly in the front legs. They can detect carbon dioxide, body heat, and they're also very good at detecting various biochemical cues. And as soon as you get close to them, they stick out a couple of legs, and if you brush up against them, they'll get onto you. They can't jump or fly or anything like that. They don't drop out of trees, but they're very good at glomming onto you if you walk by. And people have asked me, well, how cold can these things actually be out and feeding? And they're cold-blooded creatures, so once it gets below freezing, they can't be out and feeding. But I've personally collected them when air temperatures have been 41 to 42 degrees. But they move rather slowly. They get on you, but they fall off pretty easily. And if it gets a little bit warmer, then they're much better at sticking to you and finally attaching and getting a blood meal. Like I mentioned, that the adults come out as soon as the snow melts in the spring, and we were collecting them on, I think, March 2nd this year. My record in Minnesota is February 11th. I found black-legged ticks out feeding one of these years where we didn't have much of a winter. And when we talk about the habitat where we find these things, it's important to think about the landscape across the state here, because the ticks are not found everywhere. They're found primarily in wooded and brushy habitat or just outside of it. They have to be pretty close to this, and it's this type of habitat that provides the higher humidity. It's held in right at ground level and allows the ticks to survive. Again, the ticks are going to spend most of their time right down here in this leaf litter, rotting leaves on the forest floor, and on days when the humidity is high enough, they'll come up off of the ground and feed. I've researched sites like Camp Ripley in Morrison County, where I've gone to sites in the morning, and it's been nice and humid, and the ticks are up, and boy, you can collect large numbers of them. And I've gone back to the same site later in the day when the sun's higher, the wind has come up, and the ticks are extremely hard to collect then. You have to kind of go deeper back into the brush, and then you can still find a few. So they're kind of fickle at times. Unfortunately, I think there's so many of them out there that it's still not hard for people to run into them. Even if there aren't many feeding, there's unfortunately enough of them out feeding, too many of them out. And they're not evenly distributed across the landscape. Trail edges are good places for especially the adult ticks, but the nymphs also. I don't have a good slide of this, but when you talk about the edge of a woodlot, if you have a transition between woods and like an open lawn or an open field, if it's an abrupt transition, there'll be ticks there, but not as many as if you have what's called a soft edge, where you have kind of a brushy edge that's several yards wide. The more gradual that is, the better it is for ticks, I think. I certainly find a lot more in habitat like that. It's also good for the deer and small mammal hosts that the ticks like to feed on. So those kind of soft edge habitats are good for them. But you shouldn't have the expectation that these ticks are located evenly across the forest floor. They're kind of aggregated. And so when we talk about forested habitats, it's important to think about, okay, in Minnesota here, what does that mean? This is a kind of the pre-settlement vegetation here, and some of the main biomes we have. Our deciduous forest in southeastern Minnesota going through central Minnesota, that's some of the best tick habitat that we have in the state. And also that coniferous, which is the pines and spruce, that isn't the best for ticks, but there's a lot of mixed forest. There's a lot of deciduous trees, broadleaf trees mixed in with those forests now. For instance, in Pine County, south of Duluth, that's in what used to be coniferous and mixed forest, but now it's primarily aspen forest, and that's just wonderful forest for these ticks. So, and I need to do more work up in the tallgrass aspen parkland, but there's aspen forest up there that places I have sampled, I've been able to find ticks there. And the main question is how far out into what used to be prairie grassland, now it's just agricultural land, how far can the ticks penetrate out into those areas? these ticks aren't going to survive in cornfields or bean fields, but there are certain parts of the state that, even the agricultural part of the state that I think are pretty high risk, or could be high risk, especially like if you think about the Minnesota River Valley that cuts through the middle of the state. There's a nice ribbon of wooded habitat there that's just wonderful for these ticks. With the data that we've collected, we've put together various risk maps. This is the one that we have on our website now. We're going to update it soon here, but it's still fairly accurate. This is based on Lyme disease and anaplasmosis cases. They're two most commonly reported tick-borne diseases. And it's based on disease incidence, the number of cases per 100,000 residents, and so it's by where people live. Now, we also collect data. We try and talk to a lot of our tick-borne disease patients and learn about where they may have been exposed to ticks. And we're still looking at the best ways to portray some of that data, but basically it's showing the same thing, that most of our risk is in forested parts of central Minnesota, southeastern Minnesota, and kind of going out into ag land now. In recent years, we've seen a lot of activity moving northward. Since the early 2000s, some of the counties in northwestern Minnesota, like Hubbard County, Clearwater County, and Beltrami County, those have now become high risk. In north central Minnesota, Itasca County in the Grand Rapids area, that's become a lot higher risk. And we're finding scattered populations of ticks all the way up to the Minnesota-Ontario and Minnesota-Manitoba borders. My colleagues in Health Canada have found black-legged ticks in southeastern Manitoba, the southeastern corner. There's a place called Buffalo Point. It's a point that sticks out into Lake of the Woods, and they've found black-legged ticks out there. So these ticks have made their way northward. I've also been in contact with my colleagues over at the North Dakota Department of Health. They don't have much experience with ticks, but they ask me where they should be looking for them. So I happen to know some good wooded areas in eastern North Dakota, and we've been able to find scattered tick populations that far west. So I guess, bottom line is, when I talk to the public now about tick-borne disease risk, you don't even need maps like this anymore. I just say, if you're in wooded parts of Minnesota, you're probably at some risk, and it's good to take precautions against ticks, especially early on in the summer. Mid-May through mid-July is our peak risk period when those small nymphs are out. Here's some data that, or Lyme disease data, human cases, different time periods here, 1996 through 2001, 2002 through 2007, 2008 through 2013. This is Lyme disease incidence, the number of cases per 100,000 residents. The darker the counties are shaded, the higher the incidence. And you can see this spread going on here. And again, our monitoring has been done the same way, using the same case definitions and stuff over time. So we feel pretty confident that this is not an artifact of the way that we are doing our monitoring. This is something that's real. I know that I think you have this pair of maps on your website, and I wanted to talk about it for just a second. The Centers for Disease Control has surveyed public health scientists across the country to try and determine the current range of black-legged ticks across the country. And back in the mid-90s, I provided data to folks at CDC for that top map. Counties shaded in red are where the ticks were known to be established. Counties in blue is where there's records of the ticks, but not enough study was done. And then this lower map is kind of a 2015 update. And when this paper came out, it basically showed that Minnesota had the most change of anywhere. I wanted to mention, though, that when I supplied the data back in the 1990s, I was pretty strict about, there were counties that I knew the ticks were established, but I hadn't, physically collected specimens from those counties, so it was kind of a conservative view of things. Since that time, we've done a lot of work to try and identify counties where these ticks have been found. We've made it a research priority, and we're still doing that work now, but this map kind of reflects a little bit more intensive looking for the ticks too, as resources permit. I know it gets really hard to see here, but in this, I don't have a good pointer, but see the blue shaded counties here, Brown County and Blue Earth County along the Minnesota River Valley. Ticks had been reported there in the past, but just in the last few days, our staff confirmed established populations of black-legged ticks out along the Minnesota River Valley as far west as New Ulm. Next year, resources permitting, I want to go further west along the Minnesota River and see if we can document them out to the South Dakota border. Okay, as I mentioned too, with a lot of our cases that are reported to us, we try and talk to folks and learn about where they were exposed to ticks, especially our early stage Lyme disease cases. So, it hasn't been too long since that they were exposed to ticks. All of our anaplasmosis, babesiosis cases, we talk to them. What we're learning is that a lot of folks are identifying potential exposures, not only at home. A lot of people live on wooded properties, but a lot of people are exposed when they go do recreational activities in different parts of the state. A lot of people go up to their cabins in central Minnesota or western Wisconsin. A lot of people go to parks. And that's really been of interest to folks at CDC to learn more about that too, because the assumption has always been that most people are exposed at home, and out on the East Coast that's always been the main assumption, and I think that's something that needs to be re-examined there too. But here in Minnesota we've always had this, a lot of folks, especially from the metro here, that have recreational property elsewhere where they could have been exposed to ticks too. Just a couple of comments about, most of my talks are going to be about black-legged ticks here, but I want to talk just a little bit about our other ticks of importance here. We have about 11 or 12 species of ticks in Minnesota. Most of them you'll never see because they're pretty closely associated with their hosts. One of my studies used to be looking at road-killed animals. Not the most fun way to collect data, by the way, kind of messy, but there are a lot of ticks that are related to black-legged ticks that feed on specific hosts and not so much on people. Now, the real common human biters in Minnesota are the black-legged tick and then this top tick here, the American dog tick, Dermacentor variabilis. And the Dermacentor, if you see white markings, this is a female up here, she's got a white necklace, or if you see white lines going down the body, that's a male. Those are extremely abundant ticks. Historically, we considered them to be just a pest here in Minnesota. In recent years, though, we're starting to see a few cases of Rocky Mountain spotted fever, and they're the main vector of that. And there's a possibility of other spotted fever rickettsia that they may transmit. So, it's good to protect yourself against them. Nobody likes tick bites anyway, but the messages on this tick are changing over time. They're no longer entirely just a pest. They are a potential disease risk too. And then also looking for the arrival of the Lone Star tick. This is a tick that I think I mentioned this years ago when I talked to you guys too, but this is a tick that's usually found about as far north as the southern tier of counties in Iowa. But in several parts of the country, there's records of them moving northwards. They've had winter weather conditions that have allowed them to survive a little bit further north. And this is a tick that I've now seen about 30 or 40 specimens in Minnesota, which isn't much, compared to the thousands and thousands of black-legged ticks that I deal with. But most of these 40 specimens or so have been reported just in the last 10 to 12 years. So this is a tick that we think is becoming established in Minnesota. I personally collected them as far northwest as Itasca State Park. So, and as I was mentioning to Rick here before the talk, I doubt if I found the only one there. People always ask me, if you find a tick, does that mean they're established? And I think that more often than not, yes. If I find one, I'm not finding the only one. There's probably an established population. And they're a potential vector of human ehrlichiosis. And we do have a handful of cases of ehrlichiosis due to Ehrlichia chafiensis that are reported in the state every year. Most of those are in folks that have traveled to the southern U.S., but not all of them. So, I think as this tick becomes established in Minnesota, we'll have to worry about the diseases that they transmit too. Well, I should mention too with the American dog tick, they're also a potential vector of the bacteria that cause tularemia, which is a rare disease here in Minnesota, but it's definitely on our radar screen. We get 1 or 2 cases reported every year, and we have tularemia-infected animals reported every year. So far this year, we've had rabbits in Edina and a cat in Brooklyn Park reported. So, there's disease agents out there that it's good to know about. Just a few comments on tick-borne diseases. I mentioned that I'm not going to talk much about Lyme disease. You're an audience that is well aware of most of what to look for with Lyme disease, and most of the cases that are reported to us are folks that have had a history of the rash or developed late manifestations such as the large swollen joints. But I wanted to at least show a little bit of this because I'm going to talk a little bit about the other Lyme disease organism that we just identified recently too. A few more comments though on human anaplasmosis. This is a disease that we've only known about since the early 1990s, and the first few years that I followed up on cases, we'd only follow up on 2 or 3 cases a year, so it seemed like, oh, this is a pretty rare disease. Well, as providers became more aware of it, medical providers became aware, and as we started to have better testing available for patients, this is a disease that we've been getting anywhere from 600 to 800 cases reported every year now in Minnesota residents, as opposed to about 1,000 to 1,500 confirmed Lyme disease cases. So it's definitely out there, and one that we want people to know about. The main organism that we're talking about here is Anaplasma phagocytophilum. It's a bacteria that's transmitted by the black-legged tick. And just in recent years, we've identified, we worked with folks at the Mayo Clinic and have identified a new species of Ehrlichia mirus, that seems to be causing a form of ehrlichiosis in at least a few patients here in Minnesota and Wisconsin. And with both anaplasmosis and ehrlichiosis, like I said, they're related organisms, and the disease they cause is very similar, very acute onset of high fever, muscle aches, a headache, and luckily there's pretty good tests available, antibody tests. These days we're seeing a lot of what's called PCR tests, looking for the tests that look for the genetic material of bacteria. Those are pretty widely used now. There's also blood smears available where you can actually visualize the organism in a stained blood slide, but PCR is a really commonly used test. And the antibiotics that are used to treat anaplasmosis and ehrlichiosis are the same ones used for Lyme disease. As part of learning more about that new form of ehrlichiosis, I mentioned that we talk to patients about where they're exposed to ticks, and one of the things that we've been able to do is see that, interview patients in the first 20 cases here that were reported from 2009 through 2012. This is an example of the data that we put together on where people had most likely exposure to ticks. And it's in some of those same east central Minnesota counties where other black-legged tick-transmitted diseases are common too, so it wasn't too much of a shock to us. Babesiosis is kind of an interesting disease. It's a parasitic disease. This is one where we think a lot of folks are exposed to this organism, and a lot of folks are able to fight it off without any illness whatsoever, but they can be infected for a long period of time, which becomes important when we talk about blood transfusions, and I'll talk about that in a few minutes here. But for people who are symptomatic, they tend to be either folks that have compromised immune systems or elderly. With those people, they might have an illness with fever, headache, muscle and joint aches. If you look at their blood, they're quite often anemic. This is a malaria-like disease. These protozoan parasites are getting inside of red blood cells and lysing some of those cells. Testing for this disease, there's antibody, and the PCR testing is available for this too, and is used quite frequently. Blood smear is also available, but that PCR test is really good. Powassan virus is an interesting one. With a lot of these, with both mosquito and tick-borne diseases, there's kind of a list of potential agents that could be in this state, but we just haven't had the resources to look, or the resources haven't been available across the country to look. Powassan virus is a good example of this. This is a virus that science has known about since the 1950s. It's first described out in a case out in Powassan, Ontario. And there have only been a few dozen cases reported across the entire U.S. Since that time. But there's only a couple of laboratories that could even do testing for this particular virus. This is a virus that's closely related to West Nile virus and transmitted by ticks, though, instead of mosquitoes. And I thought that perhaps that we had this virus circulating here, and it wasn't until, I think, 2008, we had a case where a child was bitten by a tick and developed central nervous system disease. I was able to convince folks to send off specimens to the Center for Disease Control and the New York Department of Health, the only two labs in the country that could test for this virus. Both labs came back with positive results on that patient, and we were able to justify the creation of testing here for Powassan virus at the Minnesota Department of Health. We became the third lab in the country to test for it, and as soon as we did that, we were able to identify additional cases. So there are diseases like this that are just kind of waiting to be better described as testing becomes available for them. And similar to that Ehrlichia mirus organism, we've interviewed patients to see where they're exposed to ticks. Some of the same east central Minnesota counties. Makes sense if the black-legged tick is the vector. And I just put in Lyme disease incidence there to kind of show the darker shaded green counties are where Lyme disease incidence is high. And it makes sense if the same tick is transmitting both disease agents that some of the same counties would be high risk. And with the available resources, we've been able to test ticks from various parts of the state for Powassan virus. The counties that are shaded here, the 6 counties, are counties where we've looked for the virus. We've been able to find the virus in all 6. We haven't been looked at other counties yet, but we've, with the ease that we were able to find the virus in the 6 counties that we've sampled, it appears that this virus is pretty widespread across the state. We found infection prevalence, well, one year at one site we had 0%, but most of the time it's about 3 to 4% of the ticks are carrying Powassan virus. So, this virus is definitely out there. We think there's probably a lot of asymptomatic infections, but we don't know, the full clinical spectrum. It probably behaves like West Nile virus, where most infections are asymptomatic, but some people will have an illness with fever, and then there's just that subset that have central nervous system illness like encephalitis and meningitis and more life-threatening illness. Those are the ones that are most likely to be diagnosed because they're the ones that are most likely to be tested. And there are other emerging tick-borne disease agents that we're concerned about too. You may have heard of Borrelia miyamotoi. It's a relapsing fever spirochete related to Lyme disease bacteria. And if you look in the literature, it's an agent that's been described across much of the United States. We think that the black-legged ticks, Ixodes scapularis, are the vector of that. Recently, it's been associated with illness with fever, chills, headache. Rash seems to be kind of uncommon with this particular disease. But this is one that we need to get better human tests available. And stay tuned. I think we'll probably be describing human cases of Borrelia miyamotoi infection here in Minnesota in the future. We have described a couple of cases of this next one, Borrelia miyonii, and this is something that was in the news a few months ago. We worked with the Mayo Clinic and the Centers for Disease Control to identify this new species of bacteria. It hadn't been described before. We found evidence of it just in Minnesota and Wisconsin patients. Mayo was able to go back and look at thousands of patient specimens submitted from all over the country, and the only, these are specimens that were submitted for tick-borne disease PCR testing, and they only found evidence of this agent right here in Minnesota and Wisconsin, or people that had visited our states. So this is one that, again, only 2 cases have been identified in Minnesota to date, but this could be the next anaplasmosis for all I know. We need to get more testing in place, and as that happens, we'll see if we find more cases. As I mentioned, we do see some tularemia. It's a Francisella tularensis. That's the agent that causes tularemia, but it's a low number of human cases. and just a low number of animal cases too. Okay, so when we talk about transmission of disease agents from black-legged ticks to humans, not all ticks are infected, and not all infected ticks can transmit. The longer a tick is on somebody, the more likely it is to transmit disease agents. For the most part, Lyme disease is transmitted after a tick has been attached for 24 to 48 hours. Some people say, well, I know I got that tick off quickly, but yet I still got Lyme disease. Sometimes those folks have been bitten by more than one tick too, so that's always a concern. Whenever somebody says that they found a tick on themselves, I always tell people to check more closely to make sure you didn't miss other ticks. Like the other day, I found one on me, and then I looked a little bit more and found several more here. So they can be out in bunches and very hard to find. You might not get every one that is there. Powassan virus is the most scary thing as far as tick transmission time. We think that, there's animal studies that seem to indicate that transmission could occur in less than 15 minutes of tick attachment, which isn't too much of a shock because, again, on the mosquito side with West Nile virus, as soon as a mosquito starts to bite you, if it's infected with West Nile virus, it's transmitting that virus right away. So this is very similar on the tick side, that the ticks could transmit this virus quickly. But it doesn't give us any grace period at all, which is very much a concern. I mentioned that with our surveillance program, looking at the cases the same way over time, we can look at trends in the data. These are data from the year 2000 through 2014. If I had a graph that went back further, and I could go back to the mid-90s, everything just kind of went further down. The general trend lines for Lyme disease is in the green, anaplasmosis in the blue, babesiosis in the red. Everything is, in general, a line going upwards. There are little blips on the map, or on the graph, I should say. Like if you look at Lyme disease in 2003, there's a big dip there in the number of cases. What happened then? Well, that was a year that we had a pretty good drought conditions right during that high-risk time from mid-May through mid-July. And the ticks apparently weren't out and feeding, and if they can't come out and feed, they can't transmit disease agents. So there's conditions like that kind of factor in. 2014 was the same way for us, kind of a dry year. Our 2015 data are almost ready to release, and I think we'll be showing the numbers popping back up again. But this is what concerns me, that this trend line for everything is going the wrong way. It's going upwards. When you look at who's at risk for tick-borne diseases, here's some data by age groups. I haven't revised this slide in a while, but the numbers don't really change that much. Lyme disease we see occurring in people of all ages. That's the blue bars there. About a third of our cases are in kids. We think that a lot of that might be due to some behavior differences. Kids are less likely to check themselves for ticks and less likely to stay on trails, things like that. They're popping around the brush a little bit more. But if you look at human anaplasmosis and babesiosis, most of the cases that are reported to us are folks in their 40s and above. And everybody's being exposed to the same ticks. So we think that kids and young healthy adults, for the most part, have asymptomatic infections with both anaplasmosis and babesiosis. Not all of them. We do have some younger folks, but for the most part, the age distribution is kind of shifted. This is an American Lyme Disease Foundation slide, but it shows pretty good data on, or pretty good information on the time of year that the ticks are active. The nymphs tend to be active from mid-May through mid-July. In Minnesota here, they peak in June, and that's kind of our peak risk month. Last year, we had some kind of goofy weather, and the numbers actually peaked a couple weeks later in early July. So there's a little bit of a swing there, but in general, the spring and early summer is the highest risk period because these small nymphs are out and feeding at that time. The adults can be out early in the spring, as soon as the snow melts. Again, most years now that's sometime in March. And they'll be out through June. They kind of fade out by the end of June. You can see that males and females that I collect towards the end of June, I can tell that they're almost out of energy. They're moving more slowly. Their colors are faded. They actually look kind of weak and running out of gas. Hard to generate much sympathy for them, but they fade out. But that next generation of adults, if a nymph is successful in feeding in the early summer, it molts into that next generation of adults that comes out in the fall. They tend to peak in October, and in recent years, again, with kind of extended fall conditions, warmer weather going into the fall, we have black-legged tick activity well into November. I think December and January are the only 2 months that I haven't personally collected black-legged ticks here in Minnesota. And just to look at when disease cases are occurring then, okay, we saw when the ticks were out. This green line here and the green bars indicate when most of the cases of in this case, Lyme disease and anaplasmosis are reported. So most people are getting sick in June, July, and August. And if you kind of backtrack the incubation period of the illness, it matches up nicely with when that nymph stage of the tick is active. And we put this slide in for mosquito-borne disease risk, which is higher later in the summer. One of our messages to the public is early summer, think ticks. Mid to late summer, think mosquitoes. And then there's a little bit of a risk in the fall there too with the adult ticks coming back out. Now, I mentioned that we'd try and do some field research on these ticks. The main method that we use is called drag sampling. This is an old slide. We now wear white suits when we do this work, like you saw in the first slide there. But we have about a meter square piece of cloth, and it has little fingers on the back side that are weighted with fishing sinkers that get right down to the ground level, get down to that leaf litter. And at our research sites, we'll have transects through the woods. It might be anywhere from 100 to 750 meter transects. And we walk those transects. We can't wear any repellent or anything because we're the bait. But we walk those transects and stop every 15 or 20 meters, remove ticks off of ourselves and off of the cloth. We can get an idea of the number of ticks per 100 meters that we're coming across, so we get a rough idea of the abundance. But we also can take any ticks that we collect and bring them back to our laboratory and test them for the disease agents. It's kind of an expensive process to test them, but we test as many as we can. And here's just a real brief summary of what we've been finding in the ticks from 2005 through 2015. This represents several thousand ticks that were been tested from around the state. Primarily from 4 monitoring locations that we've been monitoring long-term. Itasca State Park, Camp Ripley in Morrison County, St. Croix State Park in Pine County, and then some state forest land down in southeastern Minnesota in Houston County. But what we're finding is that you don't have to look too far to find infected ticks. And I know that you guys all know that, but it's important to get some data behind this too. On average, over a third of the adult black-legged ticks are infected with Lyme disease bacteria, and over 1 in 5 of the nymphs is infected with Lyme disease bacteria. That varies from site to site and from year to year. We've had sites where over 60% of the adults have been infected. So, but I don't like these odds. If you have 3 adult ticks on you, one of them is probably infected with Lyme disease bacteria. That's pretty high infection prevalence. Anaplasma, Babesia, the Ehrlichia murus organism, Borrelia miyamotoi, and the Borrelia mayonii, the infection prevalence is a little bit lower, but again, not too hard to find ticks that are infected. And for somebody who's done a lot of work on mosquitoes like myself, when we first started testing ticks, this was a bit of an eye-opener because in the mosquito world, like with West Nile virus, if you get 2 or 3 mosquitoes out of 1,000 that are infected, that's considered to be a high infection prevalence. So when I saw numbers like this, is pretty alarming. So again, bottom line, you don't have to go far to find ticks that are infected with something, and quite often we find ticks that are infected with more than one pathogen too. And I know there's been quite a bit of talk about co-infected ticks and also just co-infected patients then too. I think it's important for physicians to know that if they diagnose one tick-borne disease in a person, that it's good to know, at least have it on the radar screen that potentially your patient may have been infected with another agent. Okay, just some comments on what we're doing this year for research. Again, the amount of work that we can do from year to year varies quite a bit. I work at a state agency, but similar to a lot of other programs at the Department of Health, we don't have any state tax dollars going into our program. We're basically funded through federal grants. So years where I have a little extra federal money, I'm able to do more field studies. And we've attracted a lot of good attention from CDC in recent years, and they're interested in working with us on several things. But here's some of our field studies that we're doing this year. I mentioned tick infection prevalence over time. Those 4 sites that I mentioned, Itasca State Park, Camp Ripley, St. Croix State Park, and State Forest down in Houston County, we have several years of tick infection prevalence data at those sites, and we want to keep that going into the future. What I'm interested in seeing is how much those infection prevalences change over time. You know, are we going to see things stay the same, or are we going to see a lot more ticks infected? It's kind of early to tell, but I think it's important to look at the same sites over time to see how things are changing. We're also doing a project with CDC collaborators right now on what we call the phenology of the tick life stages, the time of year that the ticks are active. At some of those same sites, at 3 of those same sites and then 1 more site, we're collecting ticks every other week during the field season, and getting an idea of when the different life stages are out. We have pretty good idea of that already, but it's good to really pin down the amount of variation that we could see here in Minnesota. This is information that helps not only to let people know about when they're at risk, but also in the future if we have area-wide control measures that are available for ticks. It's good to know where and when to go after the various life stages of the tick. We're also starting a collaborative study this year with folks at CDC looking at the reservoirs of the tick-borne disease agents. I mentioned that most of the ticks are picking up the disease agents when they feed on mice and chipmunks and other small mammals. The white-footed mouse is particularly important, but as the ticks move further north in Minnesota, they're moving north out of the range of the white-footed mouse. Once you get north of Highway 2 in northern Minnesota, white-footed mice are a lot less common. So we're interested in what other animals might be involved in maintaining these disease agents and being important hosts for the ticks. So we're starting some work on that this year. And again, that's information that might be able to help us with control someday if we target those animals for any host-targeted acaricides or something like that. We're also working with CDC on an initial effort here to look at residential versus recreational exposures. We know that some people are exposed on their properties, but where on their properties? Is it right where the woods joins their lawn? Is it in their gardens? Where on the property are people being exposed? We're starting a study in Washington County this year to look at several residential yards and also nearby recreational areas, looking at the tick densities in those areas and also the infection prevalence with disease agents. We're also trying to document the presence of black-legged ticks in undocumented areas. Those 2 U.S. maps that I showed you, trying to get records of the ticks in counties where we haven't found them yet, to further justify, get the word out to people that they should be protecting themselves in some of these counties that they may not have considered to be risk counties. Then this last one's kind of an unusual thing. On the East Coast, exotic species of all types are of interest these days. And out on the East Coast, there's exotic vegetation problems. In Minnesota here, we have problems with buckthorn. Everybody's probably heard about buckthorn. But there's another plant called Japanese barberry. It's a commonly used ornamental. And out on the East Coast, there have been a couple of papers that suggested that in areas where this barberry has escaped cultivation and taken over the understory and woods, that's ended up being really good tick habitat and may increase the risk of disease in those areas. In Minnesota, we have just a few areas where Japanese barberry is spreading outside of cultivation, and we're doing some work down in southeastern Minnesota this year to look at relative tick populations in the exotic plants versus outside. Again, hopefully to help us better target control options once they become available. A couple comments on blood transfusions. Most people get a tick-borne disease, get it through a tick bite, but not all. And I've been involved with the investigation of several transfusion-associated babesiosis cases. You can imagine if I mentioned that most people that are infected with babesia have asymptomatic infections. They have low parasite load in their red blood cells. They fight it off eventually, without illness. But if during that time they decide to go donate blood, unfortunately their blood can be transfused into somebody else. And if you think about a population of people that needs a blood transfusion, that's definitely not a population of people that you want to put a potentially life-threatening tick-borne disease agent into. So we've documented several transfusion-associated cases of babesiosis. The blood banking industry is just bringing on screening now for this particular disease agent. Different hospitals can buy babesia screened blood, and it costs a little bit more, but hopefully eventually it'll be industry-wide. We've also, with human anaplasmosis, we've documented 2 transfusion-associated cases, the first 2 ever reported anywhere. Back in 2007, 2008. We've worked on another couple since then. And with Powassan virus, that's something that theoretically could be transmitted through transfusion, but we haven't documented cases yet. So this is all information that we try to get out to medical providers so that if they're seeing a patient that, like recently we followed up on a patient that had open heart surgery in January and then developed anaplasmosis. Well, you're not going to get anaplasmosis in January through a tick bite here in Minnesota. And so, we're in the process of confirming that right now, but it looks like the patient received some anaplasma-infected blood. So it's important to think about, how these disease agents can potentially get around besides just in a tick. However, when push comes to shove, we want people to avoid tick bites if at all possible. That's where the emphasis of a lot of our work is trying to get information out to people about where and when they're at risk. That's probably the most important thing. And if people are out in the woods, they can walk in the center of trails. In this particular slide, one of my employees walking in the middle of the trail, which is a little bit lower risk. That other employees kind of off in the rough there a little bit and a little bit higher risk. This is an example of a little bit of a soft edge, though. There's a lot of brush there, and that's a good-looking trail for ticks, unfortunately. We recommend that people wear long pants and light-colored clothing so they can see ticks on themselves or have a better chance of seeing ticks. And then wearing repellent, and I'll talk about repellent in more detail in just a second. Performing tick checks is important, although I think it's tough for people to do, unfortunately. Trying to find all these little tiny nymphs on a person's body gets to be an extremely time-consuming and almost you almost have to be lucky sometimes to find them. And there are some things you can do to control ticks at home that I'll mention in a second. But with repellents, DEET is still an option, although it's not the best repellent for ticks. It will repel some ticks. Permethrin-based repellents are much better, though. For people that live in wooded areas or spend a lot of time in the woods, we get messages all the time, or comments from people saying, well, I live in the woods, I can't put on repellent every time I walk outside, and we agree that's not practical at all. But with permethrin, you can apply permethrin to your clothing. It lasts through several washings, lasts for several weeks. You can put it on, forget about it for a little while, and does a very good job of repelling ticks and will kill any that come into contact with the tick. So I think that's the most viable repellent option we have at the moment here. Comments on removing attached ticks. I think I probably showed you this slide before, but this is the business end of a black-legged tick here, the mouth parts. These two parts here are the palps. They kind of spread apart when the tick bites, and it's that hypostome, that thing that looks like a Christmas tree that actually goes into the bite site. And these ticks are kind of hard to pull out, since they do kind of burrow in deeply, as opposed to a regular wood tick. If you pull a wood tick off, it looks like there's skin in their mouth parts. That's not skin. They have kind of real blunt mouth parts, and they dig in just a little bit under your skin and glue themselves on with saliva, so it's actually dried saliva that you're seeing in their mouth parts. But black-legged ticks, on the other hand, they use very little saliva, and they dig in deeply. and it's not unusual for those mouth parts to break off when you pull the tick out. There's not really much evidence that increases your risk of disease. If the disease agent is already in the mouth parts, it's probably already in you anyway. Prompt removal of ticks is the important thing here. We tell people not to get too fancy about removing ticks. Frankly, I don't know how many thousand ticks I've removed off myself over the years. Every one of those has been just pulling them off with my fingers. The important thing is to get them off quickly. It's not unusual, though, like I said, for those mouth parts to break off, and they will work their way out over time. During the course of any summer, I usually have a few sets that are working their way out. It can be kind of irritating to some people, but it shouldn't be that much of a disease risk. Let's talk about tick control for a couple minutes here. When it comes to tick control, we don't really have good landscape-wide control options that are available yet. A lot of folks are thinking more about different methods that could be used now. And there are lots of different ways you can kill ticks. The trick is to find one that's cost-effective on a large scale. What we do recommend, though, is kind of an integrated pest management approach, same type of approach you'd use for mosquito control too, and it includes several things. The main thing is to do surveillance, know exactly where and when the problem, where the control is needed. You don't need to do tick control everywhere or be aware or take precautions against ticks everywhere. It's important to know that high-risk times and places. Public education is always a strong part of an IPM program, knowing the tick biology, knowing the personal protection that people can use to protect themselves against ticks. For homeowners, there are landscape management practices you can use to reduce tick habitat. The drier you can make the habitat, the better. So, that transition area between woods and open lawn, for instance, if you can keep the grass mowed short, that does make it tougher for the ticks to survive. Some people put landscape barriers in. I'll show a slide in a second on that. Some people even rake up leaf litter where the ticks live. That might be kind of hard to do if you own a lot of woods. And with a lot of these control measures, it's important to kind of think about where people would come into contact with woods. If you own 40 acres of woods, you're probably not walking around those 40 acres equally. There's probably trails that you walk along, or a mulch pile that you bring stuff from your garden into. Areas like that are where the emphasis should be placed for control. And when we talk about control, sometimes people talk about host animal management. For small mammals, nobody's really seriously talked about doing mouse control out in the woods. They're just too prolific to really have much of an impact on them. But people will remove mouse habitat, like brush piles and things like that, from their immediate surroundings, make it a little bit harder to have tons of mice around. White-tailed deer, there's been a lot of discussion about white-tailed deer control. And there have been people that have excluded deer from areas using deer-proof fences. And those show some promise in small areas, but unfortunately, you have to reduce the deer population to a pretty low level before you really see much of an impact on the tick population. I've seen some papers where deer populations have been reduced somewhat, but then you just end up with more ticks on the remaining deer, so it's a bit of a challenge there. Protection of companion animals is important. Not only for the animal's sake, but oftentimes dogs especially can bring ticks into a house, and if the ticks have already attached to the dog, they'll feed on the dog and stay there. But if they haven't attached yet, they can get off onto furniture and people, and before they dry out, they can be a real problem. We have several folks in that have been reported to us that have had a tick-borne disease. They might be an elderly person that lives in a wooded area, but they never go out in the woods. But Rover does, and Rover comes in the house and unfortunately brings ticks in. Acaricides, which are pesticides geared towards tick control. There are two main areas that have been investigated there. Just general applications, in other words, spraying tick habitat to kill the ticks directly, and then host-targeted acaricides, where you try and apply the pesticide to either small mammals, the mice, or the deer. Both show some promise. With the general pesticide application, again, That works best if it's done in, along the edges of trails or other areas where people come into contact with ticks, not across a huge area. And you need to deliver the pesticide right down to the ground level, which can be kind of a challenge at times because there can be a lot of low-growing vegetation that intercepts a lot of that pesticide, and maybe the ticks will come into contact with it when they come up the quest, but the control can be kind of spotty there. The host-targeted acaricides have shown some promise, at least in small areas. I saw on the back table there when I came in, there's some Daminex tick tubes, they're called. They have permethrin-laced cotton balls on the inside that mice can crawl in. They take out the cotton and incorporate it into their nesting. And then as the mice come into contact with the permethrin-coated cotton, it kills the ticks on them. There are also bait stations that have been tried commercially for small mammal host-targeted acaricides. The same bait stations that folks use in the pest control industry. Basically a mouse runs into a little mouse house and there's a piece of bait in there, and to get to the bait they have to run through some curtains that are treated with a pesticide. And if you have enough of those set up in an area, you can impact the population of immature ticks. Unfortunately, a lot of these methods cost several $100s per acre per year. So the key is going to be trying to find effective methods that are a little less expensive. Host vaccination is another thing that's been tried experimentally, actually treating bait with drugs that will kill the ticks either the ticks or the pathogens themselves, in the mice. So both those things have been tried experimentally. Nothing has been shown to be feasible on a large scale. And again, a lot of people will think about their properties. This resolution's not good on this slide, but we want people to think about their properties and think about where the ticks are living, and how they could kind of make a separation between where the ticks live, the tick zone, and other parts of their yard. Again, we're going to be studying some of this year with the CDC folks, looking at ticks and how much the ticks can get out into garden habitats or ornamental plantings, or et cetera. Some people will either try and rake up leaf litter, which is an option. It's pretty labor intensive, but it makes removes that habitat for the ticks to live in, and could be done at least along the edges of trails and make it a little less tick friendly. A lot of people also use landscape barriers, either rock or wood chips. Rock is probably a little bit drier and better. The data on landscape barriers is kind of mixed. More often than not, it serves as a good indicator that if you cross over the barrier, you're getting into tick country. But still, it makes it just that much harder for the ticks to be a few feet out into the yard. What I found in my field studies, like I said, the ticks are very closely tied to wooded habitat, but this time of year, the adult ticks especially, they might be a few feet out into the open, since they're a little bit less likely to dry out. People always ask about the Lyme disease vaccine. This is an old slide that I dug out back when Lyme disease vaccine, Lymerix, was on the market. It was on the market from 1999 through early 2002. It was pulled due to poor sales. There's a theoretical risk that the vaccine could cause arthritis in some folks, and that was enough to really kill the sales of the vaccine. We were in the process of evaluating it at the time it was pulled from the market. It had some promise, and there are some vaccines that are still being worked on in other parts of the world, especially Europe, but nothing that's going to be coming out on the market here soon, as far, at least I haven't heard of anything. One of our concerns with the Lyme disease vaccine too is that we didn't want people to think that they were invincible once they had this vaccine, since there's all the other tick-borne diseases out there. And then just towards the end here, I just wanted to mention a couple things. I get asked a lot about climate change and how that might impact tick-borne and mosquito-borne disease risk. And with tick-borne disease especially, we know that as climate changes over time, disease risks are going to change. The main question is how much and how quickly, and will it be up or down. Most of the climate models right now are predicting increased temperature, over the coming decades, which could lead to longer tick growing seasons, lower mortality in the winter, new tick species like that Lone Star tick becoming established more further north here, and with them, new disease agents introduced into an area. Most of the models also predict increased precipitation and increased humidity, which is probably as important as anything, because with that increased humidity, the ticks have more time to come out and feed. The ticks actually probably do a lot of feeding at night. They feed on mice and deer and things like that are active a lot at dusk and dawn and during the night. So there's a lot of tick feeding that occurs when we're generally not out in the woods. And if climate change leads to increased forest habitat, then there'd be more habitat for black-legged ticks. Of course, the converse too. If it gets warmer and drier, it's going to adversely impact the ticks and probably lower tick-borne disease risk. But I think most of the models right now are predicting warmer and moisture conditions. If there's more forested habitat, there might also be increased host populations, and with increased host populations, that's just more ticks that might have something to feed on and become more successful and establish into new areas more easily. Ticks are probably being introduced into new habitats all the time, and it's just when you get a critical mass kind of being introduced into a new patch of woods that find environmental conditions that are good for themselves and enough hosts to feed on, those infestations kind of catch on. But at the end of the day, our main challenge is trying to protect people from these little nymph stage of the black-legged tick. And with that, I will, take questions.
Military uniforms routinely are treated with permethrin.
Mm-hmm.
So obviously the government knows that it's an issue. Why isn't the general public made more aware of the danger and risks? For example, maybe signage or other warnings. And what is the state doing to alert people to the risk?
Yeah, well, of course, whenever I talk about tick-borne disease, and protecting I stress protection and I stress the use of permethrin these days. As far as signage goes, I'm starting to see a lot more land managers signing areas now. Some of the state parks, like Itasca State Park, has a little interpretive exhibit. Folks in Washington County this year, Washington County Parks, they have new signage up at all their parks. I think it's something that you'll see more of over time. I know that way back in the late 80s, early 90s, I had a little bit of resistance to notification from, I know like some of the chambers of commerce in different parts of Minnesota, they didn't want to scare tourists away, but hey, the tick-borne disease is pretty widespread here in Minnesota. Everybody needs to know about it.
Okay, terrific. We've been told that Zika causes birth defects because it is a visible defect. Can Lyme and other tick-borne illness cause neurological defects?
Well, certainly Powassan virus can cause neurologic damage. We know that for sure. And so can Lyme disease. If Lyme meningitis and Lyme encephalitis are less common manifestations, but if they occur, that can certainly lead to either permanent damage or some of this is a lot more evident than some of these manifestations are more evident than others. With Zika virus, having a baby that has a small head, that's pretty obvious. A subtle neurologic problem is a little bit harder for some people to appreciate. A lot harder to show on the news too. Yeah.
Good. A question here. My neighbor has over 13 people, mostly children, diagnosed last year with Lyme disease. What can they do? What is being done in that area? And that's up in the Forest Lake area.
Well, in Washington County in particular, we have a very strong collaborator in the Washington County Public Health Department. Fred Anderson, he's really interested in, first of all, gathering data on the recreational parts of areas within the county, but also learning more about what's going on with residents. And that's part of the reason we're doing our cooperative studies with CDC in Washington County this year, is because of the folks there being very receptive to working with us on learning more about exposures on properties versus recreational areas. I don't know if this particular family has woods on their property, but people can be infected more than one time, so it'd be important for these folks to take precautions against ticks and maybe talk about some of the property or the land management practices that can reduce ticks too.
Great. Dr. Malloy, did you have any other questions? Okay. Here's one. What is the process for reporting confirmed case of Lyme disease? So you reported numbers up here. What's the process for?
The process is, in Minnesota, we have a list of reportable diseases, reportable by law. And so what that means is if a doctor diagnoses a case or has a suspect case, they're supposed to report them to us. And if a laboratory has a positive test, they're supposed to report those to us. In practice, the reporting from laboratories is much better, especially these days. Most of the laboratories have automated reporting streams set up so that if there's 10 positive tests in this test run, that they do a couple hundred tests in a day and 10 of them are positive, those 10 automatically get reported to us. Much tougher, it's much tougher to get reports from physicians. And we've done studies looking at underreporting of tick-borne disease in Crow Wing County. We did a study a couple years back looking at underreporting of Lyme disease. What we found is for every case that was reported, there were another couple that went unreported. Those tended to be cases where people went to the doctor with a rash, got treated right away, and no testing was done. But once those cases are reported to us, then we investigate each one of those reports, and we have a national surveillance case definition for Lyme disease and all the other tick-borne diseases. It's kind of much maligned because it's a fairly narrow case definition. It doesn't capture everything, but the important thing is that if we look at the same case definition over time, we can see trends in time over time and space.
Dr. Maloney?
Well, and that's a good point, Dave, about how you have that surveillance case definition.
That's different from a clinical case definition.
And that's, I think, what that trips up patients from getting diagnosed earlier, and I don't know how do you communicate with physicians? Because we're still getting lots of reports where physicians seem so unclear about Lyme disease, and yet this presentation tonight is very clear.
Yeah.
Way to leverage your website better, or?
Well, perhaps. In recent years, the case definition the confirmed case definition has remained the same, but there's been a probable case definition added, and that's to kind of capture a lot of what doctors are, whatever they diagnose as Lyme disease, along with lab confirmation. But as far as getting the message out to providers, we give as many talks as we can. We do have some information out on our website. What we found is that doctors tend to be kind of a moving target. You have to keep talking to these folks time and time again, and new doctors in some of the same clinics that you talked to in years before. It's kind of an ongoing process. And I think it might be just kind of an evolution too. But we're certainly open to considering ways to talk to these folks.
What about medical school?
Medical school, I'm not sure how much training they get there on tick-borne.
Yeah. Get them before they're such a moving target.
Yeah, I know that we have a program where residents come through the health department to kind of learn about Public Health 101, and every year we talk to a couple hundred residents over the year. And, I give them a tick-borne, mosquito-borne spiel, so we get to some of them, not enough of them, but.
See, our organization, the MLA, and everyone here is really passionate about trying to determine what can they do to be most effective in raising this issue within the state of Minnesota. Is there any suggestions you have? I found it interesting that you said you get your funding from the federal government, and so none of it actually comes from the state legislature, which was interesting for me. But what can we do? So if our organization or individuals want to say, how can I be most impactful to get the knowledge out within the state, what would you advise?
Well, it's kind of a complicated question. I mean, because you almost, what we've seen in medical field and in public health in general is that whatever captures people's attention the most tends to attract the research and prevention dollars the most. And the best example of that right now is Zika virus. I'm responsible for monitoring Zika virus in Minnesota, but I do so wholeheartedly. But I also know that that's a disease of travelers for Minnesotans, and tick-borne disease is much more important to our residents than Zika virus. But if you look at the amount of media attention that generates, we saw the same thing with West Nile virus too. As long as West Nile virus was in the media, a lot of money was put into research, a lot of money was put into prevention. As soon as it dropped out of the media attention, media spotlight, it went down. Now, I say that Lyme disease is complicated because at various times Lyme disease has really been in the media quite a bit, and I don't know. I'm perplexed sometimes at the best ways to publicize things. Quite often, well, almost every spring, I haven't done it yet, but almost every spring we do a news release at the Department of Health saying, we're entering into the highest risk season for tick-borne disease. Now's the time to protect yourselves. A lot of times I get pretty mediocre media interest in those stories because it's not new to them.
Good, that was going to be my
So one of the things I do is, when we do have a new disease, pointing at my contact information, but when we have a new disease to talk about, I try and talk about that so that we can, bring up the whole topic of tick-borne disease.
Just raising the visibility,
Yeah.
that was going to be my follow-up. Do you get contacted regularly by the news media
Yeah.
to give, updates on the news, on radio, etc.? I know Dr. Maloney's been on TV before.
I do.
And is that a good avenue?
It's a good avenue, and it's a way to get messages out. Just the other day, I did a story for Minnesota Public Radio, and Lorna Benson, the reporter there, I've worked with her for years on various stories, and with her, I planted a seed. I said, hey, we have this kind of crazy way of looking for ticks. We put on white suits and drag through the woods, and she wanted to do a ride-along to see what that was like. So she did that last week with us, and the story was broadcast on NPR this week, and they have pictures up on their website. And we're trying to do what we can, but you almost have to look for new ways to capture people's interest.
Great. Another question. Bartonella was not on your list on your list of diseases.
It's on my radar screen as a potential tick-borne pathogen. We're lacking, clear evidence that it is at this point, but it's definitely on the radar screen. Some of the work that folks are doing, and we've supplied ticks to some of these efforts. There are people that are kind of looking at ticks to see what's in them, which is one of the first things that you can do to see what might be transmitting various disease organisms, or what organisms might be pathogens, potential human pathogens out there. I think one of the huge changes I've seen since 1985 is just this explosion of all these other tick-borne diseases that have been identified, and I don't think we're done yet. We're going to identify a lot more pathogens and a lot more reasons for people to protect themselves against ticks.
Another question here is, what's unique about the black-legged tick that make them more of a concern for transmitting infections?
Well, there's a number of things that are unique. I know from just a real practical or a real day-to-day observation. I do a lot of field research on ticks. I get lots of ticks on me. Black-legged ticks, they get down to business right away. They climb up, they attach, they start feeding, whereas the American dog ticks, the regular wood ticks, they'll hide into the seams of your clothing, or they'll walk around forever. I call them stupid ticks sometimes, because it's like they forget what they're there to do. They take a while to start feeding a lot of times, where the black-legged ticks don't mess around. That's one thing that they do. I think the ability for the black-legged ticks to exploit a bunch of different potential host animals is also a strength of theirs. That's something I've studied a little bit in the past, and we'll be studying it a little bit more this year too. But they utilize a variety of different mouse species and voles and small mammals. They're not picky, whereas some ticks are. Some ticks are very host-specific, Yeah.
And then you mentioned, you test ticks frequently. Do you test those in-house, or do you send those out to have them tested? What's the...
Most of the ticks are tested in-house. We have our Minnesota Department of Health Public Health Laboratory. We have a set of PCR assays to look for the genetic material of all these different tick pathogens. Last year, with some of the collaboration we did with CDC, some of the ticks have been sent down to them for testing too. It costs a fair amount of money to test every tick, $15-20 a tick, and that adds up in a hurry. And they're testing a bunch that we won't have to pay for. Now, unfortunately, we sent them a bunch of ticks, and then Zika came along, and all their lab people have been working on Zika virus instead of testing our ticks. But we'll get the results back sooner or later.
Maybe as I wait, is there another question from the group here that hasn't been covered already, and then we'll go back to some others. Yep.
I was undiagnosed for a year and a half,
Mm-hmm.
and then I was treated, and I gave blood because I'm O positive. I gave blood for several years, and then about a year and a half ago, I was in a study for post-treatment, and I found out I was never treated properly for the disease. What are the chances that I gave Lyme disease to another person because I gave blood? I worry about that.
Well,
I took my name off of Red Cross and all those.
Right.
I will not get blood.
Chances are low. Lyme disease is something that theoretically could be transmitted through blood transfusion, but usually the period of spirochetemia, the amount of time that the spirochetes are circulating in high numbers in the blood, is pretty brief. So there's kind of just a short period of time where you could transmit, could donate blood with spirochetes and pass that on. Other than that, the bacteria settle out in different tissues and aren't available in the blood that's taken for blood donation. Now that Borrelia meionii organism that I discussed, the new type of Lyme disease, that organism appears to remain in high spirochete levels in the blood. So that one might be a different story. We'll have to see on that one.
We'll do the other questions from up here. Hiking trails. You showed pictures there of. Why isn't that information as you go to your county parks, state parks? Why isn't that information available on how to utilize the trails? And there's a question here: can they gravel the trails? And I know that comes down to cost.
Cost, yeah.
But
well, and what I've recommended to DNR folks, like for state parks, is to mow trails to the extent possible and keep brush cut back. But depending on the park and the trail system, that can be a challenge. A lot of parks have pretty small trails, but people shouldn't know that they're at higher risk if they get into these narrower trails with overhanging brush.
Yeah, so how does that information, because what you showed here was really helpful, and I'm sure a lot of lay people don't realize to walk down the middle, et cetera. So is there a chance of getting that sort of information at the parks where people can realize if they're going to utilize the parks, how to utilize them in a Lyme-safe way?
Yeah, well, that's something that's happening at some parks. It's just kind of spotty at this point. And like I said, I've had kind of blanket talks to DNR folks. I've had talks with some of the county park people. But it's something that we just need to have happen a lot more consistently.
One more question?
Sure. Is there a way, since I guess many of us in this room have Lyme disease, that we can check and see if we have them reported to your database? Because I know that my doctor intended to report it and probably did. But we have no way of even telling if we're on the database or not. And that might be a data point for you.
Yeah, you can. You can call me up and I can check. I have to be a little bit careful because theoretically we need to protect not more than theoretically, we do have to protect the privacy of patient data. So sometimes we will send out a form that sign of records release or something, so that we can tell you, make sure that you are you before we say that. We have to be kind of careful. Tick-borne disease doesn't have the stigma like of HIV or something like that, but still by law, we need to protect privacy.
Great.
But we can help you out.
Yeah, this question's come up a couple times, and Dr. Maloney, I'd like you to shed some light on this. How do we get this information out to the doctors in Minnesota, and I know Dr. Maloney's been very involved in trying to do that. What would you recommend
Well,
in your role?
Yeah, we do what we can, and what that usually entails is a combination of our website, and we do several presentations every year. Last year we did, I think it was 29 presentations to physician groups, do a lot of grand rounds. Again, sometimes it can be hard to get enough doctors in a room, though, to hear all this and soak it in.
Right. And I know Dr. Miller, you've been doing things also, correct, to get information out to.
Right, so I'm doing online course modules, so they can get continuing medical education credits by just going to the website.
Okay, terrific. Any other questions from the group that haven't been covered already? Sure.
Living in Minnesota, you're obviously going to find ticks on these quite frequently. So if you, we already found 2 in our family this season, and how do you decide when to actually and when to not? Because we all know that many of us are asymptomatic.
Mm-hmm.
When do you know when to treat and when to not?
Well, yeah, I can't talk about treatment since I'm not a doctor, but what I tell people, first of all, if they found a tick, that's good news. You know, so many of the people that we talk to that have had a tick-borne disease, they never even know they're bitten by a tick, and so finding one, you know, you're already ahead of the game, and at the very minimum, you know, you can watch very closely for any signs or symptoms of illness. You know, if you find a tick that's attached to you and engorged, you know, if you take that tick off and go to the doctor, you can get prophylactic treatment for Lyme disease, a single dose of doxycycline. That's something that some people will do. If you do that within 3 days of getting the tick off of you. But really, what it is just an indicator that you can really it increases your awareness for any symptoms that you have. So many folks that we talk to will have kind of a non-specific illness. You know, they might blow off the rash as something else. They might think the joint aches are due to something else, or the fever is due to something else. You know, it's just nice if you had a tick on you to actually know it and then be aware.
What about with children? My son goes to the school on a farm and pulls 2 ticks off of them before. You can't tell me if it's doing safe. You know, like what you're doing for kids.
Yeah, with kids it gets a little bit tough. Well, kids can be easier to check for ticks. That's one bonus. There, you know, it's easier to check a little kid, and they have such unblemished skin and everything. But it can be kind of tough sometimes to know if a kid is ill. You need to look for some pretty subtle changes, and that's one of the reasons why we're just trying to get the word out to folks that, you know, if you've been out in the woods, even if you haven't seen any ticks, if anybody's noticing any kind of illness that could be tick-borne within a month or so after being out in those woods, it might be good to get that checked out.
Just a couple more questions here. According to the CDC, attached to the Minnesota Department of Health website, it states that you're developing and testing strategies for the control, prevention of Lyme disease in humans. So that's a goal. What's currently being done with respect to testing and controls?
Well, I know there have been a couple of large acaricide studies done in eastern states looking at pesticide treatments to try and kill ticks in residential settings. Other than that, you know, what's really being done is some of the groundwork. You know, some of the studies that I was mentioning to kind of get that information about where and when controls should be targeted once options become available. But a lot of the control research is also done at in university settings too.
Super. Maybe a couple more questions and we'll wrap up.
Well, you alluded to the vaccine that didn't work and was removed from the market. Where and is there anybody really working on a vaccine?
Again, I think there's a couple of European companies that are, but nobody's testing anything, for use here in North America specifically for Lyme disease.
Why not?
Well, that comes down to an economic decision, I think, for some of the pharmaceutical companies. I was amazed when Limerix was marketed. Their marketing budget was, I think, $20 million, for when they launched that vaccine. That's a pretty good chunk of money.
One more question?
I was wondering, it seems like the biggest barrier for getting treatment for Lyme disease or any of the other infections seems to be insurance companies. Do you guys work with that, or how can the public work with that? Or is that just something you guys don't deal with at all?
We don't deal with it at all, but we certainly hear those frustrations all the time.
It seems like they're telling the doctors not to, my children, I pass through every time we've gone in, it's just the doctors are like, you're not Lyme, it's just done, but it seems like it's the insurance companies that are fueling that, not the doctors themselves. The doctors seem to have their hands tied, so.
Yeah, and that's definitely a set of issues that are important to try and address it at some point, but that's not that's a completely different part of the operation than what I'm involved with. But I hear those frustrations all the time, and it's frustrating to me too, especially when I hear folks that definitely need to be evaluated and or treated and aren't getting what they need. Obviously the insurance companies are trying to save a buck where they can.
Do we have one?
I think, when you look at the economic impact of what this disease does to people, long-term chronically ill people, the cost and the disability, Social Security disability, Medicare costs, insurance costs, because people are not knowing it's Lyme, so they're being treated for all kinds of other diseases, even arthritis in children. So they're being given long-term medications that are doing more damage than good because they're not being diagnosed.
Yeah, no,
What's the economics of it?
yeah, and actually, I think Centers for Disease Control is hearing some of that concern about the economics too. One of the a study that I didn't mention here because it wasn't a field study, but we're working with Centers for Disease Control and 3 other states on a cost of Lyme disease study, and we're taking a group of 300 Lyme disease patients in each of our states. And that's confirmed and probable cases, so it includes some of the longer-term treatments that you're talking about, and just interviewing people every month and pinning down the exact costs so that we can estimate, the burden of Lyme disease on society. And the hope, I think CDC is hoping that might attract more attention to this eventually.
Can we follow that study?
What was the question?
Can we follow that study?
It's hard to follow, but eventually the results will be made public, that's for sure. Yep.
What patient I just had that, I'm so curious. What patient database are you getting your cases from?
Our surveillance database, so it's confirmed and probable cases.
But I mean, just not to be the fly in the ointment, but a lot of people will start with a doc in the community, and then for whatever reason, their treatment will halt, and the patient would think that they might need more, and so then they go to people who aren't, they're going to physicians, but who aren't, in an insurance database. So
Right.
do you lose those people, or do you still catch them?
No, we still catch them because they're enrolled, and we're interviewing them directly about where and when they're seeking treatment. We're also getting indirect costs too, the cost of missed work, or travel costs associated, anything that could be. Yeah.
And one more question?
I'm frustrated because as what you're talking about with the Lyme disease and all these pathogens that possibly be hearing, why there isn't a greater concern? I mean, how much money was just sent, billions of dollars, for the Zika virus, and this is just largely being ignored, it seems. Why is that happening? I mean, it's been 30 years and testing hasn't changed. There's no more tests. And we're seeing more things. You're seeing more infections.
Again, I think in my career, I've noticed that whatever's most visible in the public eye, public health-wise, really gets the attention, and attention equals funding. And so that's why we try and talk about these diseases as much as we can, especially the new ones, and how things have changed. I'm glad that I did a fairly conservative reporting of our tick distribution, in that back in the 90s, because this paper that came out in 2015 showing this huge change in tick distribution in Minnesota, that led to a lot of media coverage and gave me a chance to talk about how things have changed. So it's a lot of this is media driven, unfortunately.
Well, that's good. Thank you, Dave. Terrific information, great questions. It's almost 8:30, and I want to thank you so much.
Thank you.
Just to let you know, Dave works all day. He comes here on his own. He's not, constantly through the state, so we appreciate it so much.
Long day, yeah.
Dave, to come out and talk to us.
Thank you.
Thank you.
Yeah, thank you.